Hi guys, this is a really quick voiceover for an even quicker lab. This lab is lab, I think it's 7-2. Oh shoot, I should know this. Hold on. Yeah, 7-2 of your book. This isn't on your lab practical, but I did want to do a quick PowerPoint and voiceover on it simply because one, you have a quiz. Also, a little heads up on that quiz. I'm going to give you 2 attempts on that quiz, just because I do feel like this is a little bit tricky, and it's also just giving you an opportunity to get maximum points here at the end because all of you guys want those points, and you can stop bugging me about the points. And so I'll give you 2 points or 2 attempts on that quiz, just so that you can kind of take your time with it and kind of understand this process that's involved in this, because it is important if you're going into practicing medicine, and it's something that I kind of want you to take your time with and learn about in a non-pressure kind of way. So go through the quiz as treat it, that first one as a learning experience, and then treat the second one as your actual quiz. So let's talk about the antimicrobial testing. Again, this is a fun one to do in lab. This would have been on the practical had we been in person, but again, I just want to kind of go over it because this is something that's going to be important to you, and I want to talk about why that's important. So first of all, I also want to take a minute here and say, if you haven't listened to lecture, I think it's lecture 16, the lecture on antimicrobials, you should probably do that before you do this lab. I talk a lot about different concepts and definitions and the why behind antibiotics and antimicrobials that are kind of important for understanding, putting this lab into context. So if you haven't done that, pause this, quit this, and go do that, and then come back to this. So I'm going to assume you did that. And let's review why narrow spectrum antibiotics are the best. Remember that we want the antibiotic that specifically and effectively works against the kind of bacteria that is causing the problem in a patient. So we don't want, if we use a broad spectrum antibiotic, it's going to potentially lead to other side effects, toxicity, dysbiosis, right? It's going to basically wipe out our microbiome, and we don't want that. So we really want an antibiotic that's going to be very targeted towards that towards that bacteria that is causing us problems, because that will give us a larger therapeutic index, a wider therapeutic window, decreasing the side effects and the chance for leading to dysbiosis. Because remember, those C. diff infections are real, and they are big. When you get into practice, you're going to find that that's like all anybody talks about in a hospital setting, especially if that infection is systemic or intestinal. That's where we see those C. diff infections kind of rear up and cause a lot of problems. So in addition to all of that, of course, we don't want to use, by all means, we don't want to use an antibiotic that doesn't work. And so there are some very common bacteria that cause very common infections that are very resistant to commonly used antibiotics. Penicillin, those beta-lactam class antibiotics are really are commonly how should I say this? A lot of bacteria are resistant to them because they have that beta-lactamase gene. In addition, gram-negatives are very resistant to many antibiotics because they have that outer membrane, and it protects them in a lot of different ways. Okay, so we needed to come up with, or scientists and physicians needed to come up with, a standardized method for screening this. So we could do that diffusion method that I briefly talked about in lecture number 16. It had to do with basically creating lots and lots of tubes and diluting the antibiotic to see where we get that minimum inhibitory concentration. But a much higher throughput method, and therefore a much more effective method, is the Kirby-Bauer method. And this method has been standardized by the CLSI, so clinical lab standard. Oh, what is it? The Clinical and Laboratory Standards Institute, which is a department of the CDC. And I'll show you really quick. I think I can get out of here. Let me see. Okay, so for example, so here's their website, the CLSI website. And so you can actually buy the performance standards book. It's pretty expensive if you, it's nice light reading for you at nighttime. But if you're running a diagnostic lab, this is kind of like your little Bible when you're running these Kirby-Bauer tests, because it's going to give you the testing standards for all the different antibiotics that are out there, and cross-reference those with known bacterial standards. So there's again an entire book. And then what's interesting about the CDC's website is they'll put out, for example, some emerging some emerging bacterial infections that have shown some type of resistance. So the two that they have currently, I just kind of looked up their, let me see where we're at here. I looked up their most current update on meningitis issues with bacteria and antibiotic resistance. And the two that they have a current standard on is the Haemophilus influenza and the Staphylococcus pneumoniae. I'm sorry, Streptococcus pneumoniae, and what their current standards are. So you can see here's the different antimicrobials. This is the concentration of that antimicrobial that's in the disc, and then they give you those zone standards in so E. coli is the I think is the standardized. This is what you can kind of compare. It's a positive control for each of these antimicrobials. Okay. Let's go back here. And the other, so it's standardized. So in other words, what is really nice about this is that you can take, you can do this test here in San Diego in a diagnostic laboratory here in San Diego, and you can compare those results to something, a test that someone did at another point in time halfway around the world. So because everyone does it in the same way and the parameters of the test are held to a very high standard, we can now kind of we create this platform on which we can compare results, which is really nice. So before I get further into this, I wanted to, again, briefly, I talk about this in the lecture, but I also want to talk briefly about the difference between antimicrobials and antibiotics. I misuse these terms all the time. I transpose one for the other, and I just want to make it clear because I do make this mistake a lot, and people do this in general. So antimicrobial is really a term that is it's the broadest term. So an antimicrobial is literally anything that acts against a microbe. So for example, bleach is an antimicrobial. So I should say it's also a substance. It can't be a process. So for example, drying something out or putting it in the sun, those are antimicrobial actions, but they wouldn't be considered an antimicrobial because it's not a substance. So an antimicrobial would be any substance, liquid, powder, whatever, gel, that will act against a microbe. Now so this really is the broadest possible term. So they tend to be synthetic, right? Think about bleach, for example. The first antimicrobial that we found was it Ehrlich? I think his name was Paul Ehrlich. He found salvarsan, that arsenic derivative that worked against Treponema pallidum. These are synthetic. So if it's a product that's produced by an organism, probably a microorganism, a microbe, then we would now call that antimicrobial substance an antibiotic. In other words, all antibiotics are antimicrobials, but not all antimicrobials are antibiotics. Do you guys ever do these Venn diagrams? They were all the rage when I was in high school. They were like, it's like concept mapping to try to put them in your mind. I don't know, maybe they've gone out of fashion now. But I want to contrast this with, for example, another term that I think we didn't get a chance to go over. I think it's chapter 5 in your textbook. It talks about mechanisms of control. So let's contrast an antibiotic to a disinfectant, right? A disinfectant is a substance that is going to be applied to to inanimate objects, right? To the surface of an inanimate object. So if it's a disinfectant, it's going to be something that's synthetic. Ooh, is there a non-synthetic disinfectant? So is there something? Oh, well, okay. So something, just to contradict myself while I'm lecturing, it's always a good thing. Vinegar would be actually a pretty good example. I'm sure you've heard of how to be kind of more eco-friendly. You can dilute distilled vinegar, and you put that in a spray bottle and use that to wipe down surfaces. And it is broadly and mildly a disinfectant because we're applying it to a surface. But believe it or not, vinegar is actually technically speaking, an antibiotic. It's a product of a living thing. So vinegar is acetic acid, and acetic acid is a fermentation product. So if you have any hippie friends, they probably make their own apple cider vinegar, and you could basically put I forget what bacteria it is, and you give them apple cider, and you basically let it sit there and rot and smell really funky, and you get apple cider vinegar. So you can distill that into white vinegar, and you can use that as a disinfectant. So all these different terms, right? So broadly speaking, anything that acts, any substance that acts against bacteria or microbes in general, I should say, bacteria, viruses, fungi, and so on, are considered antimicrobials. If it comes from something that's alive, like penicillin, for example, comes from that bread mold, Penicillium. Streptomycin comes from the Streptomyces griseus bacteria, and so on. Then we would consider those antibiotics. Okay, so let's talk about the recipe. Let's talk about the recipe for this Mueller. I didn't even put the name of the of the agar here in the presentation. I should have. It's called Mueller-Hinton agar, and hold on, let me just Hinton-Mueller. Yeah, Mueller-Hinton agar, and it's. I was always surprised. I figured this should be a defined broth because it's such a standardized test. But basically what they do is it's undefined because they give a really broad range of nutrient sources: beef extract, casein, and starch. Make it undefined, and between those three nutrient sources, most bacteria can live and grow on this Mueller-Hinton agar. There is an extension actually of the Kirby-Bauer method where we supplement the agar with sheep's blood for those fastidious bacteria that can't otherwise survive just on Mueller-Hinton agar. But that's it's basically an extension of this test, and we don't need to get into that. Okay, so even though it's an undefined broth, there's a couple of undefined agar, I'm sorry. I wanted to point out a couple of interesting things about preparing this media, and that is that it has a very high agar concentration. So the agar, I think it's usually like 5 grams of agar per liter of water, and for Mueller-Hinton, I think it's 15. It's triple, I think it's roughly triple the amount that you would normally put in there. And I'll talk about why that is here in just a second. There's a very narrow pH range. They want to make sure that pH is between 7.2 and 7.3, I think 7.2 to 7.4. And the depth at which we pour the media into the plate is really important as well. I want to say that it's defined at 4 millimeters for a effective test. Why? Why is it so important that these things are how they are, right? Why must the plate itself be standardized? Well, let's think about our plate. So if we have a plate, so let's imagine that we're looking down, right? Our plate is on the table and we're looking down at the plate, and we're going to put this little tiny paper disc that is saturated in our antibiotic, and we're going to put that on the surface of our media. And so we can see that here if we're looking from the top down. Now, you know what? I'm going to just really quick pause this and animate something. Hold on. Okay, cool. Okay, so let's talk about the antibiotic disc first. Again, we're going to saturate it with a certain concentration of our antibiotic of choice, whichever one we're studying. And if we were looking at this from the top, we would see just this kind of little small white paper disc on top of the agar. And now let's look at this top one. Let's imagine that if you squat down and you're eye level with a table and you're looking at the plate from the side. Right? So now you can see the plate will kind of look like a rectangle, and then this little black line is representing the paper disc that's resting on top of the agar. Okay. So we've standardized the concentration of that antibiotic, and the idea here is that when we place that antibiotic disc on top of the agar, that the antibiotic itself, or the antimicrobial, is going to diffuse throughout the medium. And we don't necessarily know what concentration is where, but we do know that the concentration of the antibiotic or the antimicrobial is going to be high, close to the disc, and it's going to get less and less and less at some point. And so we don't know, does it reach all the way to the edge of the plate? Does it stop somewhere in the middle? We don't know, but we know that there is a gradient of concentrations. And we also know that because we have this media standardized, that it will be the same every time we do it. So this is why it's important so I think our standard is 4 millimeters of depth. This is why it's important. Notice that this one is too shallow. We only poured it to 2 millimeters, while this one is too deep. We poured it up to 6 millimeters. And so if we can kind of visualize that antibiotic spread right? Remember, you did diffusion in 107, right? Where the chemicals are going to diffuse throughout a gradient. They're going to move from high to low, areas of high concentration to areas of low concentration. That if we pour that plate correctly at 4 millimeters, the antibiotic is going to, and again, you can't see this. I just made it red so you can kind of visualize it. It's going to spread at a certain rate over time, over 24 hours, and then it's only going to get so far. If we put it in too shallow of a plate, that diffusion is going to go laterally, or horizontally, I guess I could say, horizontally faster, because it can't go down now, right? So our zone of inhibition is going to get much bigger if we pour the plate in a too shallow. And likewise, if we pour the plate too deep, we're going to get too small of a zone of inhibition. So it's really important that we have really well prepared plates that have the correct amount of agar. Again, think about if the agar concentration was too low, the antibiotic would diffuse throughout the entire agar very quickly, and that wouldn't work either. If it was too high, if we doubled the agar concentration, then the antimicrobial wouldn't be able to diffuse enough. So it's all about finding that range where we see an effective diffusion of antibiotics so we can determine where the antibiotic works and where it doesn't. In addition to standardizing all of the parameters of the media, right, whether it can be too deep, too shallow, if the pH is off, that could interfere with the function of the antibiotic itself, the agar concentration, and so on, don't forget that antibiotics are molecules, right? And remember that molecules have a characteristic diffusion pattern. In other words, large molecules will diffuse large molecules will diffuse slower than small molecules, right? The smaller the molecule, the faster it can move throughout the media. So if we have an antibiotic whose molecular structure or molecular weight is quite small, it's going to have a faster and therefore bigger diffusion pattern than an antibiotic that's very big. So it's really important when we're thinking about this, kind of the theory behind this test, that we don't compare antibiotics to each other because their diffusion patterns are different depending on their molecular weight. Okay, so how do we, and I also want to point out, so this is what I wanted to point out, is that even though I'm showing you one disc, right, the advantage to a Mueller-Hinton agar is that we can do lots and lots of discs on the same plate. And so this plate is actually a really big plate. So if you can remember the agar plates that we were using in our lab, they were, I want to say maybe about 3 inches in diameter. And these, if we were in a diagnostic lab and this was something that we do as a service to the hospital, we would probably be using plates that are 6 inches. So they're really big plates. And that way we can fit even more screening discs onto the plate so we can analyze lots of antibiotics at the same time. Okay, so we have the theoretical setup. We understand how the agar works. We understand how the antibiotics diffuse throughout the agar. So what we're going to do now is we're going to plate the bacteria onto the agar. And what we do, it's very simple. We simply just aseptically transfer the bacteria onto the surface, and then we just streak the you-know-what out of it. We're just going to streak it all over the plate, turn the plate, streak it again. And the idea here is that we're going to get a really nice, even, consistent lawn of growth over the entire plate. And that's what we want to see. And you can see that here, right? All the cloudy portions. We have a nice uniform carpet of bacterial growth all throughout the plate. So again, just for simplicity's sake, I'm pretending that we're only screening one antibiotic at a time. So how do we figure out if this particular antibiotic X, is inhibitory to the bacteria that I applied to this plate? Well, we have to find the zone of inhibition. So we streak the bacteria onto the plate, we apply our paper disc that's infused with the antimicrobial, we incubate for 24 hours, and we're going to see this zone of inhibition where we have growth, lots of growth outside that zone, and no growth in that zone. And it should be a really nice uniform halo around that paper disc. So a couple of terms here, right? Our zone of inhibition is going to be the diameter, not the radius. So we're going to measure exactly across at the biggest point of the circle, and we're going to measure that in millimeters. And that's what we're going to consider our zone of inhibition. And that's the data point that we want. That's the piece of data that we're going to compare to our table in the next slide. And then at the end of that zone of inhibition, right, so that edge, where the bacteria is going to start to grow, this is the minimum inhibitory concentration. So anything, let me, hold on a second, I'm going to get my pen. Okay, so if we were to look at this point versus this point, right? Bacteria can grow. I'm sorry, they can't grow here. The concentration of the antibiotic is enough to stop the growth of the bacteria there. But it's not enough here, right? So that border between where bacteria can grow and where they can't grow is called the minimum inhibitory concentration. And remember that the antibiotic is probably everywhere, right? It's diffused throughout the media. And it's high here, right? It's higher, I should say, in the zone of inhibition versus outside the zone of inhibition. So we don't really know where the antibiotic is. The only thing that we do know is that whatever the concentration is, we don't even know the concentration. We can't say, it's 10 micrograms per microliter or something. We don't really know what the number is. We just know that on one side of that zone of inhibition, it can't grow, and on the other side, it can grow. Okay. So this is where we take that piece of data and we compare it to our table. So if you want to grab, if you have your lab handy, if you want to look at table 7-3, this is going, excuse me, this is how you determine susceptibility or not. You're not looking at, believe it or not, you're not looking at how big that circle is. A lot of people think, oh, the bigger the circle, the better. And I think that broadly speaking, of course, that's true, but you also can't, you have to apply it to the table. You can't compare like two different bacteria or two different antibiotics to one another because they're going to have different diffusion patterns. They're going to be effective against different bacteria in different ways and so on. So you have to be very careful about the limits of interpretation. So let's just take an example here, right? So if we were to measure this zone of inhibition for this antibiotic, and let's just say that our ruler says that it's 16 millimeters across, we would have to compare that zone of inhibition to the table. And so if you look at your table on page 449, 449? Yeah, 449, you'll see that it can be intermediate for, chloramphenicol, intermediate for ciprofloxacin, and trimethoprim. But it's susceptible to streptomycin and tetracycline and completely resistant to penicillin. So you can't just say one size circle fits all. You have to apply this particular piece of data, that zone of inhibition in millimeters, to all of the different antibiotics that are in your table of reference. Okay, so the last thing that I want to talk about is the effect of the antibiotic itself. Again, from lecture 16, we know that antimicrobials can either be bactericidal, which directly kill the bacteria, or bacteriostatic, which stops the growth of that bacteria. So in order to figure out if that antibiotic is bactericidal or bacteriostatic, what you're going to do is you're going to antiseptically transfer. Basically, you take your loop, your inoculating loop, and you're going to run it along the surface of the area where it looks like there's no bacteria. But remember, there is bacteria there. It's either dead or not growing, right? We just can't see it because it's too small. We can see all of this bacteria out here because we have that nice, beautiful carpet of bacteria that's outside the zone of inhibition. So we're going to take our wire inoculating loop and we're going to run it along the surface here, pick up anything that's there, and we're going to inoculate that onto a new plate. And then after 24 hours, we're going to check for growth. So if we have growth, that means, of course, that it was bacteriostatic only, and that it was only preventing that bacteria from growing. If we have no growth, we know that bacteria or that antibiotic is bactericidal and in fact killed the bacteria that was within that zone of inhibition. Okay, that should give you all the information that you need to take the quiz, and also just kind of give you a good appreciation for the Kirby Bauer method. There's also a really nice, I think I'm linking to another YouTube video. He actually kind of walks you through measuring the different discs and comparing the disc to the table as well. So you might want to take a look at that. I think it's only like 8 minutes long. So, all right, that does it. Good luck on the quiz.